Hello everyone, good morning. It's my very great pleasure to introduce our wonderful speakers to you. So the first speaker is Dr. Shalima Nair, and she'll be talking to you today on genetics and epigenetics research and why it matters. Shalima has completed her PhD in genetics and molecular biology and has been working in the field of epigenetics for the past 5 years. She's been actively involved in several projects like the biomarker discovery and validation for breast cancer and studying the epigenetic variations between healthy and obese individuals. Today you'll hear from her about why medical research matters to each of us, and she'll also talk about the work she's being part of in the Epigenetics lab. So would you please welcome Dr. Shalima Nair.
Today I'll be talking to you about genetics and epigenetics research, why it actually matters to each one of us, and I'll talk to you a little bit about the research process, and then I'll introduce you to the field of epigenetics, and then I'll talk to you briefly about the work that I have been involved in the lab. So most of you here, I'm sure, would know Sir Isaac Newton, one of the greatest thinkers of our time, who discovered that Earth has got gravitational pull. But during his time, which was in the 1700s, he must have also been got thinking about all these infectious diseases that were prevalent then. And in the 1900s, when slowly medical research started coming in, we saw that smallpox had got slowly eradicated. But diseases like scarlet fever did not spare the life of Thomas Alva Edison, nor did it spare the life of Albert Einstein's daughter. But in the 2000s, things were much better because medical research was fully in on swing, and most of these diseases were controlled. But we still have complex multi-headed diseases like diabetes and cancer that needs to be tackled. So if you look at the life expectancy of man, in the Neanderthal age, which was about 30,000 years ago, the life expectancy was only about 30 years. And then in the Neolithic period, it was 38 years. And then in the Greek era, it was again not very different. It was about 35 years. And in the medieval period, which was when there was plague and epidemics, it was still about the same, about 38 years. And then in the Victorian era, which is up to 1900, it was about 40 years. But after 1900s, when medical research was beginning, there has been a huge jump, a significant increase in the life expectancy of man, which is to 75 years. And today, we enjoy a life expectancy of about 85 years. And if you look at the world life expectancy map, Australia enjoys one of the highest life expectancy of about 85 years. And we think it's because of the better healthcare system here and the good medical research and, of course, our lifestyle. So occasionally when you get sick, you do go to the doctor, get a prescription, and then you go to your friendly chemist and buy your pills. But have you ever wondered where these pills come from? How did these pills get there? So the pill has actually gone through a long journey, which is called as the research process. And what you see in the form of the pill is actually a full-blown tree with fruits in it. So the journey of the pill actually starts in some lab as a small idea by some young investigator, which is the discovery process, where this idea gets discussed within other scientists and they try to do experiments to prove the hypothesis. So they design experiments and they perform experiments on cell cultures and animal models, which is the next phase, which is a preclinical testing, where it's done on both animal models and in vitro on cell cultures. And then it has to go through phase 1 clinical trial, where it is then tested on healthy volunteers, humans. And then it goes through phase 2 clinical trials, and then it has to go through phase 3 clinical trials, and then finally it goes to approval and manufacture. So as you can see, it's a long journey. So it takes more than a decade for it to reach the stage of the pill. And Garvan plays a very important role right at the bottom, the discovery and preclinical testing, and we also do phase 1 clinical trials. So as Andrew said, the Garvan research areas, we work on immunology, neuroscience, osteoporosis, and bone biology, about which you will soon hear from Dr. Paul, hearing research, diabetes, and cancer. And when you come into Garvan, I don't know if you have seen this beautiful staircase, or maybe you will see it soon. But this staircase is here for a reason. It actually represents the genetic material of human body, which is the DNA, which is a double helical structure. And this, every cell in our body has got 23 pairs of chromosomes, and each of these chromosomes is made up of many, many, many genes. And each of these genes, they consist of DNA, which is made up of just 4 chemical letters: adenine, thymine, cytosine, and guanine. So these are the 4 chemical letters, and they are called as nucleotides. And we have in our body, every single cell has got about 6 billion nucleotides. And if you imagine each of these nucleotides to be the size of a button, which it's not, but if you imagine it to be the size of a button, and if you keep it all together, aligned together in a beautiful chain, it's going to be so long that it will cover the circumference of the earth 2 times. So it's really long. And if you imagine each and every cell in your hand, just your hand, each and every cell to be the size of a grain of sand, your hand is going to look the size of a bus. So the task that's before us is enormous. So to first understand diseases, we need to first understand all these different genes in our human body. We need to map out all these nucleotides. So a big effort was taken in the 1990s, where a huge collaboration from many international scientists, they came together and they came up with this human genome project to create the genetic blueprint of the human body. And they were successful. It got completed in 2001, but it costed them a lot of money, more than $3 billion. And it took 10 years to bring out the first human draft of genetic blueprint of the human body. But today, with the advancement of technology, you've got this new sequencing machines which can actually sequence the human genome in less than within a day, and for about $1,000 US. And the technology has advanced so much that you are actually close to the research questions. So far I've been talking to you about genetics. Now I'll introduce you to epigenetics. Has anyone heard about epigenetics? No. Yes, some yes. That's good. I'm happy. So in epigenetics, it means features that are actually on top of or in addition to genetics. So you have genes, but there are something on top of these genes. And epigenetics was considered as one of the greatest discovery of our time a few years back. So just to give you an example of what epigenetics is, George Clooney, if he was to go to a professional meeting, if he would be wearing a professional attire like this, and he would behave very professionally. But if he decides to go to the beach, he would look like this, and you know how he's going to behave. And if he decides to go to space, he would be in this attire. So something similar is happening with epigenetics. Depending on our lifestyle, the kind of food we eat, the environment we live in, there will be chemical modifications on top of our genes. So in George Clooney's example, as he wears the professional attire, he behaves professionally. But as soon as he wears the beach attire, he would behave differently. But that is not a stable one. He can change his behavior as and when he wants. But in epigenetics, it's a much more stable change. So these chemical modifications that sit on top of these genes, they don't actually alter the composition of these nucleotides. But by just sitting on top, it would actually regulate the function of the gene. Just like how George Clooney, when he wears a professional attire, he behaves differently. But in epigenetics, it's a very stable change. So this change, these modifications are more stable and can be inherited across generations. Another classic example is of identical twins. They share the same genetic makeup, but if you knew any identical twins, you would know that they have different characteristics. And this can be attributed to epigenetics. And a very classic experiment that was done a few years back, where you can actually see epigenetics in action, is that of a agouti mice experiment. Agouti is a gene in mice which, when expressed, would make the mice yellow and obese. So what the scientists did was they took a group of pregnant mice and they fed one group with normal mouse diet and the other group with the normal diet plus supplemented with compounds that are rich in methyl groups, which can actually add in these chemical modifications. And what they found was the mice that were fed with these supplements predominantly gave rise to brown and healthy mice, and the other group, which was fed with normal mouse diet, they gave rise to, again, yellow and obese mice. But the brown and healthy mice, they had these chemical modifications on the agouti gene, which actually suppressed the expression of this gene. So because the expression of this gene got suppressed, these mice no longer had yellow color, or they were not obese, but they were in fact healthy and brown. And this got transmitted to the next generation as well. So they gave offsprings that were again healthy and brown. So this is also seen in normal and cancerous cell, where you have these chemical modifications at certain regions of the cancerous cell at a much higher percentage. And the good thing about this field of research is that there are technologies which can actually identify these chemical modifications in blood, and these can be used as biomarkers for the diagnosis of disease. Now I'm going to introduce you to the lab where I work. We are a team of 25 scientists, and there I have my supervisor, Professor Susan Clark. She's an expert in the field of epigenetics. And we work on different aspects of epigenetics. So now I'll talk to you briefly about two of the projects that I have been involved in the lab. One is a breast cancer project, which is a collaborative project between Govan and University of Queensland and University of Newcastle. So we have taken part in the discovery and validation phase of this project. And we have used tumor samples from triple-negative breast cancer patients and matched normals. And we have performed whole genome sequencing of these samples. These are actually fresh frozen samples or paraffin-embedded tissues. So it's really hard to get DNA from these samples. We get very low amounts of DNA. And then we sequenced all of these samples. We had about 19 tumors and 6 normals, and studied them, and we have identified a panel of genes that are epigenetically modified, and they show strong association with overall survival. So these chemical modifications on these genes, when they are at a higher percentage, those people have a shorter survival rate. So we have seen this process. So these could be potential biomarkers for prognosis, but we still need to do a bigger validation on a much larger independent cohort, and this is what is currently in progress in our lab. And the next project that I've been involved in is to study epigenetics in diabetes and obesity. So there are two kinds of fat depots in our body. One is a subcutaneous fat, which is a fat that you see just under the skin and on your thighs. This is considered generally as a beneficial one. And the next is a visceral fat, which is a fat that you see surrounding your internal organs. And this is considered as a bad fat. And the more visceral fat you have, the more you're prone to risks of metabolic diseases. So when we started the project, we had the following questions in mind. So what makes most of these subcutaneous fat good and the visceral fat, why is it bad? Is it because are there differences, both genetic and epigenetic, in their origin? Or does these differences come in after they go and settle down in these depots, like behind the skin surrounding the organs. So where does these differences come in? And are there any genetic and epigenetic signatures that are specific for lean, healthy individuals versus obese or obese diabetic individuals? So if there are any epigenetic signatures, we can use that as potential predictors for obesity. So for this, our study design, we collected fat samples, both visceral and subcutaneous fat samples, from lean and obese individuals with a BMI for lean from 18 to 26.9, and for obese 35 to 45. We had an age limit of 30 to 59 years, and our exclusion criteria, as you can see, is smoking less than 10 years, not to be pregnant, no significant medical history, no inflammatory disease or history of malignancy. And most of these patients came in for elective abdominal surgery, so we had to take the fat out during the surgery. So after this, what we have done is we have done for 5 healthy individuals and 5 obese individuals. We have collected these fat samples and we have performed whole genome sequencing of all these fat samples, and we have got all the sequencing data, and our bioinformatician is currently looking at it systematically and analyzing it. So the idea is to create a reference epigenome map of a healthy individual. But I don't think it's 5 is a good enough number, but this is just a start for us. And if we see any chemical modifications that are distinct or specific to obese individuals, then that could be potential predictors for obesity. So, and then our next phase of this project will be to look into diabetic individuals. So this is just started, and we will have to go ahead and do the next set of validations as well. So hopefully we see the light at the end of the tunnel. But as I said, we have to go a long way to get there. And I think we will get there. Thank you so much.